This protocol supports the characterization of the role of mosquito genes involved in a variety of physiological pathways, including, for example, insecticide resistance and malaria parasite development. The method drives transgene insertion into single pre-characterized genomic locations, and therefore allows to directly compare phenotypes resulting from alternate transgenes, effectively avoiding the issue of positional effect.
This method can be applied to a variety of insect species of public health and agricultural importance, and its applications extend widely, from bacteria to mammalian cells.
Begin by designing attB donor plasmids carrying the dominant fluorescent marker, attB recombination sites, and the desired transgene cargo. Use a single attB site for integration of the whole plasmid carrying the transgenic cassette, or 2 inverted attB sites for cassette exchange. Purify donor and helper plasmids using an endotoxin-free purification kit. Combine the attB-tagged donor plasmid carrying the transgene of interest and the helper plasmid carrying the integrase to obtain a mix with a final concentration of 350 nanograms per microliter of the donor plasmid and 150 nanograms per microliter of the helper plasmid. Precipitate the DNA by adding 0.1 volume of 3 molar sodium acetate and 2.5 volumes of ice-cold 100% ethanol.